In this lesson, we'll take a look at how the action potential travels down the axon. In our previous lesson, we took a look at how the potential was generated. Now we want to see how it travels, and this is known as the propagation of action potentials. We'll finish up the lesson by taking a look at different types of axons. There's actually three different types. They're not all the same. So this is what we're calling the types of nerve fibers. Remember from a previous lesson that an axon is just another name for a nerve fiber, or a nerve fiber is another name for an axon. So propagation basically is going to move the action potential along the axon in a series of steps. There's two types we're going to look at. We're going to take a look at continuous conduction first, and then the second one will be saltatory. So let's actually skip the words for a moment and let's go to a picture. So again, if you remember from a previous lesson, that the cell membrane here of the axon is normally going to be positive on the outside, negative on the inside, and then when a neuron is at rest, we call that resting membrane potential, the value is minus 70 millivolts, right? So that's just the review. So let's just say we're at the initial segment now of the axon. Remember, that's the first place on the axon that generates the nerve impulse. So let's say we got the action potential at the initial segment. So we know that sodium is going to flood into the inside of the cell, making the inside of the cell more positive. And if you remember, that upward swing of the action potential is called depolarization, and we get all the way up to plus 30. So this is happening at the first segment of the axon. What happens now is once the sodium floods in, the sodium, if you remember again from a previous lesson, is going to move laterally. That lateral movement is what we call a local current. So if you watch these sodium ions moving over here, they're going to slowly make the axolemma here slightly more positive. Notice we went from minus 70 to minus 60. If you remember, this is threshold. Once we reach threshold, this section of the axolemma will fire. And watch this. We go to section here. I'm sorry, the next picture, right? Now this sodium floods in, right, making the inside more positive, and we go all the way up to plus 30, right? So we've depolarized this section of the axon. Now these sodium ions flow laterally, right? You can see here from section 2 or segment 2, they're going to flow laterally. And as the sodium comes over here, they're making the inside more positive. Once we get to threshold, right, the sodium is going to flood in and this cycle will repeat itself. So what's basically happening is the initial segment fires first. That change in voltage now stimulates the next section of the axon to fire. And then that fires, and again, the sodium moves over, causes a change in voltage, and now the next segment fires. So what we're getting is a step-by-step depolarization of every section of the axon as it flows down. Right? So every section of the axon is going to fire, and that's what continuous, excuse me, continuous conduction is. Okay, this only happens in unmyelinated axons. It does not happen on axons that are myelinated. They're going to get the other type of conduction. So just to go over this real quick, this affects one segment of the axon at a time. Right? Step one, the action potential develops at the initial segment. It's always going to start at the initial segment, and the membrane is going to depolarize all the way up to plus 30. Right? As that sodium is inside the cell, that sodium now moves laterally, producing that local current. We had talked about that in a previous lesson. And that now depolarizes the second segment. An action potential now happens at the second segment, and the 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 sodium continues to move in and then moves laterally and then depolarizes the third segment. As the action potential is traveling down, the first segment is going to be repolarizing, going back to normal, right? That's the downward curve. And the continuous conduction will spread all the way down the axon in this manner. The cycle just repeats itself. Roughly, continuous conduction is about 1 meter per second. Okay, so about, figure about 3 feet per second. That's how fast it's moving. All right, this is actually from another source where you can see again the initial segment here. So we're getting, again, the local current, the local sodium flowing to the next segment. That causes depolarization a little further down, right? And then it goes down a little bit further. So it just kind of moves down the axon step by step. Notice, again, these axons have no myelin on them. The second type of conduction is saltatory, right? Saltaire means to leap or to jump, like to skip. And this is going to occur in myelinated axons, and this is much faster than continuous conduction. Um, the myelin is actually going to prevent the continuous conduction, and what we're going to get is the depolarization at the nodes of Ranvier. If you remember, the node of Ranvier was also called the node. So we're only going to get the action potential where the Schwann cells don't touch, and you'll see it will leap down down that myelinated axon. Let's take a look. So again, we're going to start here. This first section will be the initial segment. So again, we stimulated it. We got depolarization happening, right? Sodium's coming in. How do I know I got depolarization? Look at the meter, right? It's plus 30. So that means that we have the upward swing. But now check this out. As this first section has depolarized, we get the local current. The sodium is going to move laterally, but the myelin is preventing depolarization. So the local current will continue a little bit further until we get to the first bare spot. Notice this is a myelin here, this is a myelin here, this is the spot in between. This is that node of Ranvier, or just the node. So now as the sodium flows here, it's going to raise the inside of the cell to threshold, right, minus 60 or minus 55. And at that point, this section is now going to fire. Let me just make this a bit smaller. Right, we go to the third slide here. Right, so once we got to minus 60 or minus 55, we reach threshold. Now sodium floods in, goes all the way up to plus 30. Now we have depolarization over here. The first section is now going through repolarization, right? It's going through the downward swing, right? So I didn't want you to think I'm ignoring repolarization. That's happening as this flow of ions continues more laterally. So now the second section has depolarized. Again, we get the sodium flowing through the axoplasm, and now it's going to go to this third segment, get it to threshold, and then this will fire, and then the sodium comes in, and then it continues down, and the cycle will repeat itself. But it looks like the action potential happening like at the node of Ranvier, then it jumps to this node of Ranvier, and then it jumps to this node of Ranvier. Right? These middle sections here that are myelinated, they're not going to fire. Okay. Um, So the depolarization only occurs at the nodes, or if you just want to change your nodes, right, to the node of Ranvier, same thing. Right, so it's jumping down. Okay, and that's again a much faster process than what we saw over here, right? The continuous conduction is much slower. The reason is every section of the axon had to go through depolarization and repolarization. Here in saltatory conduction, it's only happening at the nodes, right? The node of Ranvier. Much quicker. Okay. The last thing that I want to go over in this lesson is that Not all axons are the same, right? There's actually different types. We're going to divide it into three types, type A, type B, and type C. So the axon diameter also affects the speed of the conduction, not just the presence of myelin or the absence of myelin, but it also has to do with the diameter. Watch this. The larger the diameter, the lower the resistance and the faster the speed, right? So the larger the diameter, the faster the speed. Let's take a look at the three fibers. So the first fiber is a type A fiber, right? These are myelinated and they have the largest diameter. So as a result, these travel the fastest. Look at this, 120 meters per second. Before, when we did the continuous conduction, it was 1 meter per second. All right, so this is a lot faster. Uh, where would we find type A fibers, right? We find these in nerves supplying sensory information such as position and balance, right? So this information is going to your central nervous system from your different receptors in your body, telling the brain your position and your balance. And so a lot of sensory nerves are myelinated type A fibers. When we send out a motor response, excuse me, to skeletal muscles, those are also in type A fibers. Think about it. If you were to touch something hot, let's just say this was a spoon on the stove, right? You pick up the spoon. You wouldn't say, "Oh my goodness, this is hot. Let me drop it." Right? What happens when you touch that spoon? It's like boom, right? Your hand is like pulled away so fast. We want the fastest signal getting to your muscles to pull you away to protect you, right? So motor impulses to skeletal muscles also travel along type A fibers. Type B fibers, they're also myelinated, but these are more middle diameter. They're not the biggest, they're not the smallest, they're mid-size, and they trans information at intermediate speeds. Again, roughly 18 meters per second. These we find sending signals from the central nervous system back and forth to our organs. All right, so the communication between your organs and your central nervous system don't have to be quite as fast. So these are type B fibers. The third type of fiber is the slowest fiber. These are the unmyelinated, right? No myelin on them, and the smallest diameter. And this is what we were looking at before. They transmit about 1 meter per second. And these also send up sensory information, but they just send it a little bit slower. So you have sensory information being sent to the brain and spinal cord over A fibers, but they're also being sent over C fibers a little bit slower. Okay, so those are our three types of fibers. Okay, the last slide in this lesson, messages carried by the nerves are routed according to their priority. Critical information, right, think about it, is transmitted through type A fibers. We want that information to get to the brain as fast as possible or away from the brain as fast as possible if something is critical. Sensory information about things that threaten survival, we want that information getting to the brain as fast as possible. So they're going to travel over type A fibers. And then motor commands that prevent injury, we also want our muscles to contract. Like I said before, touching a hot stove or something, um, we want to pull away from that as quickly as possible to prevent injury. Okay, so that concludes this lesson on how the action potential is propagated along the axon and then looking at the different types of nerve fibers or axons, which also have an impact on the speed.